Hi, I'm Ellie. I'm a PhD student at Monash University, and today I'm going to present some of the research I've been doing for my PhD, where I've been exploring the relationship between geographic range size and speciation in honeyeaters. So I'm really interested in speciation, as understanding speciation can provide insight into the mechanisms underlying the biodiversity patterns we see around us today. Now, one long-standing idea from throughout the literature is that there could be a relationship between a species' geographic range size and speciation probability. This is based on the assumption that if speciation is to occur allopatrically, so through the formation of geographic barriers, a larger range simply represents a greater area in which barriers may form. However, in saying that, extremely large ranges also have the potential to engulf barriers, and therefore it's been hypothesized that perhaps speciation rate peaks at smaller to intermediate range sizes. So to test these ideas, I decided to focus on the honeyeaters, which are a large monophyletic group of birds containing about 190 species that have evolved within the last 25 million years. They have historically been confined to Australasia and currently range throughout Australia, Indonesia, South Pacific, and New Zealand. As a result, they have a huge diversity of niches, range sizes, as well as morphological traits, making them a really interesting group to study speciation. So my aim here was to test the hypothesis that speciation rate peaks at small to intermediate range sizes in the honeyeaters. So to test the relationship between range size and speciation, we also need to acknowledge the fact that range size itself is a complex trait, and a species' range is likely determined by a whole variety of other factors. Two common traits thought to influence range size are dispersal ability and body size. Dispersal ability is thought to influence a species' range size, as how far a species is physically able to move sets a limit on its range. Body size, on the other hand, has been debated a bit more throughout the literature, with some studies showing negative relationships and other studies showing positive relationships. Nevertheless, there is thought to be a relationship between body size and range size, and on top of that, body size is known to influence dispersal ability in birds, so it's an important factor to consider here. We also need to keep in mind that dispersal ability and body size could also influence speciation rate in their own way as well. The other factor I decided to include here was also range position, as position can ultimately constrain a lot of these variables. So for example, when it comes to range size, range position can influence a species' range size. So if a species is restricted to a small offshore island, their range size is likely the same size as that island, whereas that same barrier just doesn't exist for mainland species. Position could also constrain dispersal ability and body size, and on top of this has the potential to influence speciation rate in its own way as well. So to test all of these ideas, I needed three key things: a phylogeny, range data, and trait data. For my phylogeny, I made the decision to reconstruct the honeyeater phylogeny, and I used an approach where I combined nuclear genes, mitochondrial genes, and implemented topological constraints from previous phylogenomic studies. This approach enabled me to obtain a species-level phylogeny containing every single species of honeyeater. For my range data, I used expert range maps from BirdLife International and extracted information on range size and position. For my trait data, I decided to use a common measure of dispersal ability in birds called the hand-wing index. This is just essentially some wing measurements, which numerous studies have shown to be very highly correlated with dispersal in birds, and therefore is a very common metric to use as dispersal ability. For body size, I just used the typical mass in grams, and all of these values was extracted from throughout the literature. So all of this information was used in a whole range of analysis, and today I'm going to present the results from my phylogenetic regressions, my phylogenetic structural equation models using STAN, and also my trait-dependent speciation extinction analysis to ultimately show which traits were actually influencing speciation. So when it comes to range size, we see that this varies hugely across the honeyeaters. This isn't too unsurprising and follows typical species range size distributions, where in a given assemblage of species, you'll find that the majority of species have small ranges and then few have very large ranges. The honeyeater with the largest range was found to be the singing honeyeater, which spans nearly all of Australia and as a result has a range size of over 7 million kilometres squared. The species with the smallest range was found to be the Rotuma marzamella, which is restricted to Rotuma Island north of Fiji, and as a result has a range of 42.4 kilometres squared. So just a little bit of a difference. My phylogenetic regressions revealed a number of interesting relationships. When it comes to range size, I found that this was strongly influenced by position, where it decreases with longitude and increases with latitude. This latitudinal effect follows Rappaport's rule, which states that ranges are thought to be smaller towards the tropics. Interestingly, in honeyeaters, I found that range size decreases with body size. I also found that dispersal ability was influenced by range position, similar to how range size was. Here I found a weak negative relationship with longitudinal midpoint and a positive relationship with latitudinal midpoint. From here, I then decided to build a phylogenetic structural equation model, which enabled me to test all of these variables and their influence upon one another in a single model as depicted here. So I was able to test the influence of latitudinal midpoint, longitudinal midpoint, and I also decided to include a binary character of whether or not species were classified as an island species or a mainland species, and the influence of these variables upon dispersal ability and body size and ultimately range size as well. So this essentially confirmed a lot from what I found from my phylogenetic regressions. So here I see that range size increases with latitudinal midpoint and decreases with longitude, and unsurprisingly ranges are found to be larger on the mainland than they are on islands. Here we also see that range size decreases with body size, and the same influence of position upon dispersal ability, which increases with latitudinal midpoint and decreases with longitude. From here, I then decided to plot spatial patterns of species richness and speciation rate. So I took a background map and divided this up into grid cells of 100 kilometres by 100 kilometres. For my species richness, I overlaid all of the species ranges and summed up the number of species that fell within each grid cell. Here we can see that the areas with the highest species richness are the east coast of Australia and in particular New Guinea. When it comes to the speciation rate, I overlaid all of the ranges and summed up the mean speciation rate for the species that fell within each grid cell. We can see that in comparison to my richness map, this is almost an inverse pattern, where you can see that the mainland areas are a darker blue, indicating a relatively slower speciation rate in this context, and in particular, some of the islands are lighter colours, indicating intermediate to higher rates of speciation. So from here, I then moved on to my state-dependent speciation extinction models. So the way these models work is you can test the influence of a binary character upon speciation rate estimates. And I tested every single one of my traits, and the only things revealed as significant were range size and islands. So I found that range size at a threshold of 100,000 kilometres squared influenced speciation rates, with those with a range smaller than 100,000 kilometres squared had higher rates of speciation than those species that had ranges greater than 100,000 kilometres squared. To give an idea of how large this actually is, I've got a map here labelled with the areas of some of the larger land masses, and I would say 100,000 kilometres squared is slightly smaller than the North Island of New Zealand. We can also see that this covered about half the species having small ranges and about half as having large ranges. This significant result was also supported by hidden state models, in which my model, where I said that speciation rate was influenced by small and large ranges, outperformed every single one of my null models. For my null models, I included 4 character-independent diversification models, which consisted of 2 CID2 models and 2 CID4 models. I also tested the influence of intermediate ranges, and here these were classified from 10,000 kilometres squared to 1,000,000 kilometres squared. What's really interesting is I did find that this influenced speciation, but however not as I predicted. So intermediate ranges actually had lower rates of speciation than those with the smallest and largest ranges. And again, this was supported by my hidden state models. The other factor revealed as significant was islands. So mainland was classified as Australia, New Zealand, New Guinea, and Sulawesi, and islands was everything else. Here I found that islands had higher rates of speciation than mainland species, and again, this was supported by my hidden state models. So looking at these 3 factors, which we know influence speciation rate, we can see that there is an overlap in some of the species identified within each category. This could imply that perhaps this pattern is driven by one or two key clades. However, in saying that, there are a number of mainland species that have small ranges, as well as the smallest and the largest ranges associated with higher rates of speciation, still implying that perhaps range size is playing a role here. So my main conclusions are, I did find strong evidence that geographic range size influenced speciation, where I found that small ranges, as well as the smallest and the largest ranges, have higher rates of speciation. However, at this stage, I did find a significant effect of islands, so I can't say that it's independent from that. So I'd like to thank my supervisors, Stephen and Matt, as well as the Chown Lab for all of their support. And I also wish to acknowledge the people of the Kulin Nations, on whose land this research was undertaken. I pay my respect to their elders, past and present. Thank you.